This is a production of Cornell University.
So I put this slide in here just sort of as a summary slide, and basically you have two different materials here. Up here is a high CN ratio, down here is a low CN ratio. And you've seen these graphs before. This is when I showed the CO2 evolution and decomposition. Okay, so this black line here, up here and here is basically the CO2 evolution. Okay, basically it represents the activity microbial community. Okay, it's their respiration. As it increases, the population is more active. Okay, and we usually associate that with a larger population. Okay, granted it could be one really large microbe, but okay. The green line that you see there, here, right here, and right here, that's the CN ratio of the material. So here it starts very high, here it starts fairly low. Okay? This green line that's underneath the green shade, that basically represents the amount of nitrogen that's released into the system. Okay? So as that nitrogen comes down, there's less nitrogen in the system. Now they're looking at nitrate, but nitrate is one form of nitrogen. Okay? As that line gets down, the available nitrogen in the system is depressed. As it goes up, the available nitrogen is increased, right? Okay, so if I start with a high CN ratio, fresh added material right here, you can see my population just boom, but you can also see my nitrate being depressed. Now this is the nitrate that's in the soil, not the nitrate that's in the nitrogen that's in the CN, not in the plant material that they're eating. Okay, so they're harvesting this nitrogen from someplace else, the soil. Okay, as time goes on, the population decreases, the CN ratio of the material also decreases, and somewhere down the line, that nitrogen that's in the soil, that's available nitrogen, starts to increase again. The populations are crashing. Okay, they're dying. They're releasing their nitrogen. On the other hand, low CN ratio material, population does the same, bop, comes up. Okay, but you can notice because the CN ratio is so low, the nitrogen is basically waste material in the carbon in that food that they're eating. They don't need that much. Remember this. Okay, if my CN ratio is too low, I have to be blowing off CO2. I have to be making ATP, all these other things. Okay, I have waste material. And that waste material is represented in the increase of the nitrogen. It's just available. Now that's available to all the other organisms, the plants, that are in that system. Does that make sense to everybody? Now, I want to make an addendum here, and I think I've already actually made this addendum. The reality is that this is not living in isolation. Okay? The CN ratio is not, we've portrayed it sort of as it's like by itself, it's an individual control, but the reality is that if this is my population of organisms, here, this is a good one. I'm not sure I've got the right numbers, but if E. coli, you guys know what E. coli is? Microorganism. It's a gut organism, but it's everywhere else. Okay, if E. coli were left to their own devices and nothing ate them and they never died, because of their ability to replicate themselves I think within a week or something like that, some really unbelievably small time, within a week, the entire solar system would be occupied by E. coli. Something like that. Maybe it's a month or something, but it's really this small number. So that gives you an idea about how the lifespan of these E. coli, if they were immortal. Okay. But the reality is they're not immortal. And then most of the time, of course they die of old age, but most of the time they're being eaten by something because everything's a food for something, right? Okay. So if we look at this, here's that fresh, you've seen this one before. Okay. Here's that fresh material that's been added. This is the CO2 evolution, basically looking at the population, okay? But that population drops, right? So what's happening in that population? Okay, that population is doing the decomposition of the CN material, right? Okay, but if we actually look at the amount of nitrogen that's being released from the soil that's being decomposed, or no. I add fresh material, and I just have microbes in it. Okay, they're going to produce a certain amount of biomass, and they're going to release a certain amount of nitrogen. They're going to mineralize a certain amount of nitrogen. Okay, and that's what these lines basically represent here. This is mineral nitrogen that's in the soil over periods of days. Okay, this dark line right here is with nematodes. This dashed line right here is with no nematodes. Okay, and you'll note that without the nematodes, there's a lot less release of nitrogen into the soil system. Okay, so if I did not have nematodes, this is how much nitrogen would be released in the system. With nematodes, this is what's being released. Now, nematodes are sort of predatory little worms type things. And basically what they're doing is they're eating this population. And in the process of doing the eating, they're breaking them up, and they're sloppy eaters just like us. And that nitrogen that's in the biomass of the microbial population is being released. So it's not simply a relationship of the decomposers and their waste products. It also has to do with the whole trophic structure of the soil, has a huge influence on how much nitrogen is actually released. And the take-home message of the slide is, don't just look at CN ratios. You also have to look at other things. Okay, placement being one of them. Okay, does that make sense? Okay, questions so far? Cool? All right, we're going to take the last section, and that's basically litter quality. And the truth is that CN is part of litter quality. Okay, but we pull it apart because it's an important feature by itself. Okay, so litter quality. Okay, here's those CN ratios up at the top. CN ratio greater than 20, I'm having N mobilization. CN ratios less than 20, I'm going to have nitrogen release, mineralization, right? Make sense? Okay, well, let's take a look at CN ratios of a variety of different materials. Okay, the rule of thumb is high CN ratio is going to mean that I'm going to have N immobilization. Low CN ratio, I'm going to have N release. But just because I look at those numbers, I also need to look at the structure of the material is being decomposed. Okay, this stuff up here, these are low CN ratio stuff. This is high CN ratio. This is time on this DRAC, and 0 here represents the point at which above I'm seeing mineralization, below I'm seeing immobilization. Below immobilization meaning it's going into the biomass. It's being taken out of the system. Mineralization meaning the nitrogen is being released into the system for everybody else. Okay? Now I have 4 different materials here. This is straight out of your book. I have a low lignin and phenolic materials, low CN ratio. I have high lignin and or polyphenolic materials with a low CN ratio. I have high lignin and low lignin with a high CN ratio and a high CN ratio. Okay? Look at these green lines, and they basically, this is time. So the quicker it moves up, the faster it happens. Okay? And the distance here is basically representing how much mineralization or mobilization occurs. So just because you have a low CN ratio doesn't necessarily mean, this is the class of these 2 lines here are what we generally would think of. Okay? This is low lignins. This is low lignins as well. Lignin content, the ability of the material, basically represents, or the structure of that material, basically that represents how fast it in fact can be decayed. Okay, so if I have I don't know if I still have this. Did I keep this? Remember this slide? So if we think about the material and how fast it decomposes, in general, this is not necessarily true. In general, we think of the glucoses being simple sugars. They're going to be easy to decompose. The lignins being very complex molecules, being very hard to decompose. Okay? Now, go back to this slide. Low lignin on the outside, fast to decompose. The inner 2 have high lignin contents, and so as a result, they take longer to decompose. Yes, they both have the same CN ratio as these guys, and they, in general, end up at the same point, but it just takes a different time scale for them to get to that point. Does that make sense? Yeah? Now, if we look at a lot of different materials, I don't know if, can you guys actually see this slide? It's in the PDF that you guys have. But basically what you're looking at here is a variety of different plants, okay, and it tells you what the plant parts are. But what it shows here is lignin versus polyphenol content. So higher numbers, this one has a really high number, has a fairly high number of polyphenol, and basically it shows you the CN ratios. Okay, this is lignin. Its CN ratio is 28:1. This has a very small amount of lignin, basically 7%. It has a ratio of 43:1. Okay, now this is this constant is a decomposition constant. Okay, the higher the number, the quicker it decomposes. Okay, so looking here at this 13:1 material, it decomposes very quickly. But these 43 and 42:1s compared to the 28, these guys decompose a lot quicker than this. So it's not the CN ratio alone, it's the structure of the material. Does that make sense? So in essence, this is the data that filled in this chart. Okay? Now, we've talked a lot about placement, CN ratios, size, litter quality. What is it, if we're looking at decomposition, what is it that actually is, when you look at everything together, what is it that's controlling those rates? It really is the property of the organic material, where it happens to be, the CN ratio of it, the surface area of it, and its resistance to decay. Now, the truth is that all of these combined, it really is where it is located. It's location, location, location. But it's this combination of different things that controls its decomposition.
This has been a production of Cornell University, on the web at cornell.edu.